name's Molly, and today I'm going to show you how to set up an anaerobic biodegradation experiment using a procedure I helped to develop. This experimental setup is simple, economical, and can be performed by students interested in determining the potential for biodegradation, toxicity, and biogas production of various materials. We use simple, common laboratory glassware, and the experimental setup is visually monitored over time. Students will discover the complexities of determining biodegradability, anaerobic digestion, stoichiometry, and a new method for determining the fate of materials in anaerobic environments. Anaerobic biodegradation is the biological breakdown of a material in the absence of oxygen. The two main products of anaerobic degradation are carbon dioxide and methane, which are referred to as biogas. Anaerobic biodegradation can be beneficial if the produced biogas is collected and used as fuel for heating or electricity production, or as a feedstock for biopolymers. However, if left uncontrolled, this biogas contributes to greenhouse gases. Before we show you the procedure and how to monitor the experiment, you will need anaerobic seed material, the sample, which we want to biodegrade, and the setup, which includes glassware and other supplies. First, we will start with the seed material, also called biosolids, which is the active biomass. We obtained anaerobic biosolids from a digester at a local wastewater treatment plant for our seed material. However, there are many different seed materials you may use, including anaerobic sediments and landfill cores. When storing the seed material, it is very important to use a plastic jug and to keep the cap slightly ajar. This allows proper venting and has no adverse effect on methanogenic activity. Next, we need the sample, which is the material we want to biodegrade. Today we are comparing a petroleum-based plastic with a bioplastic. For quickly biodegrading materials such as our bioplastic, we use 0.07 grams of the material. For adequate biogas production, you may have to vary the size of your sample. Make sure you record the sample weight. Now that you have a seed and a sample, you need the supplies for the setup. The basic supplies for this setup consist of a graduated cylinder with a plastic guard ring, a plastic rim, a one-holed rubber stopper, and a cup of water. 50 milliliter glass graduated cylinders are used. Cylinder bases are labeled in numerical order for convenience. We use a one-holed rubber stopper with a Teflon tube through the center. Plastic guard rings are placed around the 50 milliliter line and taped into place. A wide plastic rim is placed on top. Now that you have everything needed, we're ready to start the experiment. We wear proper lab attire when setting up our experiment. This includes safety glasses, closed-toed shoes, lab coats, and gloves. Active biomass will produce some gas without the addition of other materials. It is important to include samples that contain no investigated material, called control samples or blanks. Set up these graduated cylinders using the exact same method, and verify that the cylinder is empty before you pour in the biosolids. Place the samples into the correctly marked graduated cylinders. Care should be taken to avoid getting the biosolids in one's mouth or eyes, as anaerobic digester biosolids may have an unpleasant odor. The experimental setup should be performed in a well-ventilated laboratory. The anaerobic biosolids are removed from the temperature control room, vigorously shaken, and filtered using a sieve. We have found that diluting our biosolids works well. In our case, we use 1 part anaerobic digester solids, and 1 part warm tap water. The desired amount of biomass is quickly poured into the graduated cylinders. The microorganisms present in the anaerobic biosolids are ideally exposed to minimal oxygen during this experimental setup, so it is best to work carefully and swiftly to avoid potential oxygen influences. Securely place the stopper into the graduated cylinder. Careful when inserting the stopper. If you accidentally cover the hole, the microcosm may become pressurized and biomass may squirt out, or your glass cylinder may break. It is very important that biosolids do not leak out the lip of the graduated cylinder. If you are concerned that there might not be an airtight seal, you may place a small amount of vacuum grease into the lip to prevent leakage. Covering the hole in the stopper, the graduated cylinder is placed upside down in a cup of water. The support ring ensures that the tops of the graduated cylinders, now flipped upside down, will remain uniformly submerged in water, with the plastic rim preventing the cylinder from being too far submerged and from resting on the opening of the stopper. The samples are placed in the desired temperature control room or incubator. Over time, the biosolids will settle, and you will notice clear liquid at the top of the cylinder and dark, settled material around the bottom of the stopper. Biogas will be produced from the decomposition, and this produced gas will force fluid out the bottom hole of the stopper. Students take daily gas readings using the markings on the graduated cylinder. It is ideal to have at least 1 gas reading for every 5 milliliters of biogas produced. When 50 milliliters of biogas has been produced, the experiment is complete. If you do have equipment available, you may obtain a gas headspace sample. The graduated cylinder is removed from the cup of water, and keeping a finger on the hole in the stopper, a gas-tight syringe is carefully inserted in between the rubber stopper and the glass graduated cylinder. In this case, the gas sample is inserted into a gas chromatograph, and after running calibration standards, the relative amounts of carbon dioxide and methane are obtained. The gas is analyzed to determine the carbon dioxide and methane composition. Once the gas production has reached a constant value, and a gas sample obtained if you had the equipment, the microcosm may be opened. The pH of the remaining biosolids is measured. pH strips may be used, or a pH meter if available. If the investigated material is a partially biodegraded solid, remnants can be collected. This sample is rinsed, dried, and weighed to monitor the sample weight loss. An estimate of biodegradation can be determined by the sample weight loss, the biogas production, and the ultimate methane capture. Increased understanding of the anaerobic biodegradation potential of materials will improve green chemical design and promote awareness on the need for sustainable carbon cycle. Join me in designing new green materials while educating the next generation of engineers and scientists to design for the future with environmental fate in mind.